In our series of basic courses, we will continue the basic course of immunology. And we are, last lectures, we were focusing on major histocompatibility complex, that is MHC. So today we will continue the next part of this chapter, that is MHC class 2 antigen presentation pathway. So before starting this one, first we will see at the end of this lecture what you will be able to learn. So first is what happens when pathogens enter into our body. How peptide fragments from invading pathogens are produced and loading to MSC class 2 molecules, and how MSC class 2 molecule peptide complex is presented to T cells. This is very important, which we are going to learn actually, how MSC class 2 peptide complex is presented to T cells. And lastly, we will see what is meant by the term clonal selection. So before starting our next lecture, first we will look for that what we have learned from our last lecture. So in our last lecture, we have discussed about the class 1 MSC molecules. And we also learned that, if you remember the last lectures, and if you're interested to learn this whole phenomenon, so please you can see our last lectures related to this chapter. So if you look for that, how the pathogens enter into the body, and then how all the processes, as you can see here, they goes under the viral protein, goes the digestion by proteasome, then we get into the peptides, then goes for the combined with the MSC class 1 MSC molecules, and then ultimately presented to the T cells, and then of course T cell joined with that presented molecule. So that was the story. If you are more interested, of course you can consult our previous lecture. But what we are going to talk today, we are going to focus on class 2 MSC molecules. Before going to the class MSC molecules, what you have to even recall again before proceeding our today's lecture. So if you look for that, MSC class 2 molecule present extracellular or exogenous antigens to T cells. So it means in our the last lecture, in the case of MSC class 1 molecule, we discuss endogenous antigens. But in today's lecture, what we are going to talk about, that is extracellular antigens, exogenous antigens, how they are presented to the T cells. And then in the next step, what we will learn, what we have to memorize to learn our today's lecture. So that is MSC class 2 molecule are expressed on antigen presenting cell. If you remember the last lecture, what we mentioned that MSC class 1 molecule are present or expressed on the nucleated cells, but what in case of the class 2 MSC molecule, they are expressed on antigen presenting cells. And even we also mentioned that these antigen presenting cells are also known as the APCs, and we even categorize into two groups. One we call them as a non-professional APCs, and other we called it as a professional APCs. So if we talk about the professional APCs, these are the APCs such as macrophages express class 2 MSC molecule constitutively. The example of professional APCs, which we are going to talk in our today's lecture in detail, like a B cells, macrophages, and dendritic cells. So if we go further, so first we'll look what happens when pathogens enter into our body. So in that case, we already know that where the pathogen is coming, that is coming out of the body. That could be the bacteria, that could be kind of the viruses, it could be some kind of toxin material. So what happens actually when that pathogens enter into our body? So that's going to be interesting, and how our body encounter with those foreign pathogens. So that is interesting, not only in case of the viruses, even in case of bacteria, even some toxic material. So how our body respond against those kind of pathogens. So as the MSC class 2 MSC molecule present exogenous antigens to T cells, and pathway starts when APC internalize the pathogens. So what we have to do, let's have a quick look on entry of pathogens into our body and their internalization by the APCs molecule. So what happened, if you look for here, let's supposing here, these are the pathogens, and when that is entered, of course the tissues got injured. And so in that injury, and then from that, of course, that area, other pathogens entering into the body. So what's next steps going to be here? If you look for here, these are the mast cells. What they do, why they are here. And in that is very important to remember here okay, they release a certain kind of chemicals, what we call here is the histamines. So why they release, it means first thing this is coming to encounter with this pathogen, that is the mast cell. And what they are releasing? They are releasing the certain chemicals, what we call them, they are known as the histamines. So mast cells, what they do, they can detect the injury into nearby cells and release histamine, initiating the immune response. So what are the mast cells? The mast cells are located at the boundaries between the tissue and external environment. For example, at the mucosal surfaces of the gut and lungs, in the skin, and around the blood vessels. So, and then what is the function of these histamines? And what they are doing? They are releasing the histamines. So next, of course, the questions come to our mind, why they release these histamines. So if you look for these histamines, what they actually do, these histamine dilate blood vessels so that more neutrophils and macrophage come to the infection site. So as you can see here, and they're releasing, and now these histamines coming toward the blood vessel, and they're dilating the blood vessel, and from here, if you can see, these neutrophils are moving toward the that injury part. So in that, this is what on the function, what we call, if someone asks you, okay, why the mast cell release the histamine, so what we can give the answer, to histamine dilate blood vessel so that more neutrophils and macrophage come toward the infection site. And these endothelial cells of capillaries become sticky for leukocytes, which lead toward the leukocyte infiltration. What does mean by the infiltration? It means the leukocyte could pass from these blood vessels and then they move toward the tissue, that part where the pathogens comes or where the injuries has been taken place. So that is what we can understand from here. So it means the pathogen is coming, mast cells releasing the histamine giving the message to the moving toward the blood vessels, and they're releasing to the neutrophils and macrophages toward that pathogen. So this is what actually going on here when the pathogen entered into our body. And while blood cells move in the blood vessels into the blood or infected tissues, as we can see here. So even we can summarize, as you can see here, these parts: entry of the pathogen in the body, release of the histamine by the mast cells and leukocyte infiltration, and phagocytosis of the pathogens by the neutrophils. Okay, if you look for here, sometimes these pathogens, what they do, if you look for here, they engulf those particles and they also phagocytize them, and then also try to kill those pathogens. So which type of leukocytes which reach at the site of the infection? So if you look for that answer, the neutrophils are the front line soldiers against infectious agents, and they lead the first wave of host defense to infection or tissue damage. So this is very important. The question is asked in several points and several number of different type of equations if you are a immunologist. So we must have to remember that which type of leukocyte first reach at the site of infection. So answer is neutrophils. But now another questions come to our mind, why neutrophils? So if you look for that, why neutrophils? The neutrophils are the most abundant type of leukocytes. Neutrophils are the most motile type of leukocytes, and another reason is they are chemoattractive factor of the neutrophils or produced firstly. So now we'll see, after entering and after going neutrophils towards the infection place, now what happens next? So it means what these neutrophils do. If you look for the function of and what these neutrophil does, if you talk about single one, if the number of the neutrophil of course goes toward the infection place. So neutrophils send the signal to the monocytes. Monocytes, of course, the other kind of the leukocytes which are present inside the blood vessels, as you can see here. And what happened, what they do, why they need, because they need a stronger partner. They send the signal to their more stronger and harder than themselves to send them the signal to come here, and we should fight with that kind of the pathogens. So then what happened, these when the signal getting from the neutrophils, and then these monocytes start migrate from bloodstream, and then from here when they move from bloodstream, so what happened next, if you look for here, then that after entering from outside of them, now these in case of the monocytes, they are doing, they are differentiate into the macrophages. Macrophage, it means actually the large cells which can engulf even more number of pathogens quickly, and more efficiently as compared to the neutrophil itself. So what happening here? Monocyte migrate. These are the monocyte. If they're inside the blood vessels, we call monocytes. When they goes out of the blood vessels, they are acting as a they just slightly differentiate, and then they can engulf the material easily. So they are known as the same as a monocyte, but when they start doing that function, they are known as a macrophage. And the site of the infection where they start phagocytizing the pathogens, then they of course start engulfing the pathogens here, as you can see here. So what we are going to talk the next, what happened when one macrophage take the one external molecule, one antigens and one pathogen. So what we are going to learn that, what happened the next. So this is an interesting point what we are going to learn in the next one. What we will do, let's zoom that macrophage to understand antigen processing and presentation by class 2 MHC molecules. So by zooming this same molecule, we are taking the only one example. Of course, it's not only one, lot of molecules, but to understand that phenomenon, we are just zooming one of the molecule. And after zooming, what the first process take place, that is a formation of endolysosome. So first we have to learn that how endolysosome actually formed. If you look for that in terms of the endolysosome, so what happening, the engulfing pathogens are digested with the help of proteolytic lysosomal enzyme to form a short peptide fragments. So initially what happened, when APC internalize the pathogens, if you look for here, pathogen is coming inside the body, and you can see here, this is a lysosome. And then what happening, when APC internalize the pathogens, it internalizes in the form of intracellular vesicle known as the endosome. And then that endosome, what happening to next, which fuses with the lysosome, and now as you can see for here, it is the endosome and we have a lysosome. So now what they happen, they got fused to each other, and when they fuse to each other, now that particular structure, what we call it now as a endolysosome. So I hope what happening with the endolysosome, how the endolysosome is formed. So how we can simplify answer this questions, when an APC internalize a pathogen, or it internalizes in the form of intracellular vesicle, which is known as a endosome. And when that endosome fuse with the lysosome, what is called, in that stage, that is called the endolysosome. So what we have got so far, it means after entering into the cells, after zooming, after macrophage, so what we got, and that become the endolysosome. So what that endolysosome does, if you look for that endolysosome, it does the digestion of that internalized pathogens into peptide fragments. So after the first process, what we get the second one, that is a kind of peptide fragment. So now after entering the pathogens, that has been digested by endolysosome, and now what we have a peptide fragments. So now what happening next? So now if you look for that, of course, after that we talk about the MHC class 2 molecules. And if for that purpose, the same story you have to memorize in case of MHC class 1 molecule. So similar to the MHC class 1 molecule, class 2 molecule also synthesize at rough endoplasmic reticulum. But here this is slightly different from that one. After that, then this rough in case of class 1 MHC molecule, they move toward the Golgi complex. But here they have slightly further modifications before going toward the Golgi complex. So what is that modification? So MHC class 2 molecule are assembled in rough endoplasmic reticulum, and here with the class 2 MHC molecule, a third polypeptide known as the invariant chain is associated. So what is happening here? We have a kind of invariant chain, if you can see here. So that invariant chain also combined with the class 2 MHC molecule. But the now question raised, why that invariant combine in case of the class 2 MHC molecule? So in order to block, why the answer of this question is, in order to block the peptide binding cleft so that the cell peptide did not get bind to it. So it means what they bind, why they bind, the reason is to avoiding the binding of cell peptide, or particularly like endogenous antigens could not bind because of that invariant chain. But now collectively that class 2 MHC molecule, and when it has that invariant chain, then that is known as a MHC invariant chain complex. So now what we have a invariant chain complex, and now that move toward the Golgi complex. So this MSC, this complex is transported toward the Golgi complex, and the MSC class molecule got off from the Golgi complex, as you can see here, and then what happening? They form a vesicle, and then now we have the form a vesicle, that complex. But why we are calling a complex? Because it has a, what we called it as invariant chain and also MSC class 2 molecule, and collectively what we are calling that structure as a MSC invariant chain complex. So I hope what we have to memorize in difference in case of class 1 MSC molecule and class 2 MSC molecule. So class 1 just synthesize and transport to other Golgi complex. But when we are talking today for the class 2 MSC molecule, it's synthesized at rough endoplasmic reticulum, but before going to the Golgi complex, it has this particular structure, what we call it as a invariant chain. And now we don't have only, we are not calling it as only class 2 MSC molecule, we are calling as a MSC invariant chain complex, and that move toward the, transported toward the Golgi complex, and from the Golgi complex, of course, then it moves toward, then now what is going, we already got it. What we have, if you remember our previous step, so we have a peptides. So it moved toward the peptides, or which what we called it didn't call those peptides. Only a, what we call it here, only a small fragment of invariant chain remain associated with the class 2 MSC molecule, which is known as a clip. And it, later on, like it will move toward other peptide. So it, what is happening with that one? That only the small peptide is left, other part is going to remove. So the vesicle containing a class 2 MSC molecule with a clip fuses with the vesicle containing digested peptide fragments. So now what will happen? These peptide fragments will start joining with that class 2 MSC molecule. So if you look for the next one, what happening here? Now what happening here? The vesicle containing the class 2 MSC molecule with clip fuses with vesicle containing digested peptide fragments. So digested peptide with the class MSC2 molecule, as you can see here. And now the clip is replaced by the peptide fragments, and now this MSC peptide complex is transported to the cell surface for the presentation to the T cells. So now as you can see here, they are on the surface for the presentation to the T cells. The binding of peptide fragments to MSC class 2 molecule, and they come to the surface to present this peptide to the T cells. So T cells of course can go further processes. So now we will see the next one, the antigen processing, and now we'll see the presentation by class 2 MSC molecule. So what happening here, the antigen presenting cell with processed antigen display on their surface reach at the lymph node. After that, now what happening, they moving toward the lymph node, and each lymph node has a unique and very specific receptor that is pre-programmed to recognize and react with a unique or specific antigen. This is very important to remember. It means now what happening, after processing the antigen, after processing the foreign pathogen particles and changing into their fragments, and then move toward the APCs molecule surface. After that, what happening, those APC molecules migrate toward the lymph node. And why they are lymph node? Because each lymph node has a unique and very specific receptor that is pre-programmed to recognize and react with a unique or specific antigen. And now, as you can see for here, and there is also, actually the interesting thing is here, each T lymphocyte has a unique and very specific receptor, but react with a unique specific antigen. If you look for here, they coming for here, and macrophages with the class 2 MSC molecule tries to fit in presented antigen to T cell receptor. If you look for here, come to this one, but is not recognizes here, then come to another molecule, and similarly it goes to several number of molecules, and ultimately it find the one where it got fitted. So once it get fit into the particular T cells, that clone for the T cell is selected. So it means now what we are called that, that T cell was selected for that particular antigen, and that phenomenon, that process, what we called it as a clonal selections. It means a molecule is coming, it's not selected by this one, so this is not a clonal selection, this is not selected here, but look for here, it got selected here, and once it get fit into particular T cell, that clone of T cell is selected for further action, and this process is known as a clonal selection of T cell. So I hope, okay, we should understand that if someone asks you, okay, what does mean by the process or by the term clonal selection? So process of selection of one clone of lymphocyte bearing matching antigen receptor is known as a clonal selection. So I hope, and these are very important terms. So once the antigen fit into the now, and what we call them, we know that, okay, what the receptor we called it. And if you remembered our last lecture, what we called that receptor are known as a T cell receptors. And these T cell receptors, shortly we call them as a TCR. So it means what happening here, if you look for that process, so what happening here, on the antigen is fit into the TCR, and T cells confirm the presence of MHC molecule. So what they do, if you look for here, so first they know that they are fit to that one. And now what happening? So of course T cells understand that MHC molecule have a foreign pathogen, so then it not with processing it go the same way. If we remember our whole stories in the previous lecture, so what will happen next? So after once the antigen is fit into the TCR, T cell confirm the presence of MHC molecules with the co-receptor. So what will happen in here? So co-receptor will come, and in terms of MHC2 molecule, we know that T cell co-receptor is known as a CD4. So now that CD4 bind to beta domain of MHC class 2 molecules. So I repeat, and that is the same story which we have already discussed earlier. So same things will happen, and T cell co-receptor CD4, what will happen? It will bind to beta 2 domain of MHC class 2 molecules, and thus T cell interact with the both. Now we can see that T cell has interacted with, what does mean with the both? It means with the MHC molecule as well as the antigens. So it will combine with the both MHC and antigen on the surface of the APC, and that is what we call this APC molecule. So now we can see that it is already at both process, it is on the surface of this one. But the interesting thing is here, if you go further, before going to the end of the lecture, we have to memorize one thing. Okay, how, if you look for the CD4 8 cell in the case of MHC class 1 molecule, and if you look for here in the class 2 MHC molecule, so they are specific for the CD4, while the class 1 they are specific for the CD1. So now how they are restricted? If you remember the last lecture, even I mentioned one term. Before going to the end of the lecture, we have to memorize the one term again. Okay, why they are selected and why they are only restricted to 7. That process we called it as a MHC restriction. So we will slightly again we will revise the term what is meant by MHC restriction. So MHC restriction refer to the ability of T cell to recognize a peptide antigen presented in the particular MHC molecule. Okay, which kind of MHC molecule is representing? Whether that is the MHC class 1 MHC molecule or whether that is a class 2 MHC molecule. So depending upon those one. So cytotoxic CD8 cell are restricted to class 1 MHC antigen presented by the nucleated body cells. Similarly, if you look for here, there's a story because why they're restricted, there's a phenomenon, and that process, that restriction we call it as a MHC restriction. So MHC is defining them. Okay, what defining them? Okay, cytotoxic CD T cells are restricted to the class 1 MHC antigen presented by the nucleated body cells. Why? While helper CD4 cell are restricted to the class 2 MHC antigen presenting by the APC molecule. So as you can see here, and now why they are restricted? Because of the MHC molecule have a particular kind of phenomenon, and that phenomenon is known as a MHC restriction. So if someone asks you what does mean, how they are restricted, why it mean, what does mean by the restricted? So why CD8 cell only can bind with the class 1 MHC molecule, and why CD4 is only can bind with the class 2 MHC molecule? So because MHC molecule have a restriction, and that process is called the MHC restriction. And how we can define that process? The MHC restriction refer to the ability of the T cell to recognize a peptide antigen presenting with the particular MHC molecule, whether that is MHC class 1 MHC molecule or whether that is a class 2 MHC molecule. So next what we have after presenting to the T cell. So next question raises how these T cells become activated. So therefore our next lecture related to that one. So before going to the next lecture, we will just want to go once again toward the summary. So what we have learned in today's lecture, if we just overview our whole today's lecture, So what we have discussed, APC internalizes the pathogens in the form of endosome, which fuses with the lysosome to form the endolysosome. So first step was formation of endolysosome. After the endolysosome, what happening? That proteolytic enzyme from lysosome digests the pathogen to the peptide fragment. So that's what we have already understood. And then what happening? Then MSC class 2 molecules are expressed and assembled in RER, rough endoplasmic reticulum, where they got associated with the invariant chain at the peptide binding cleft. And after that, that MSC class 2 molecule are transported to Golgi bodies from where they bud up as a vesicle. And now what happening with those vesicles? We already understood that these vesicles fuse with the endosome and peptide fragments are loaded to MSC class 2 molecule for antigen presentation. And after that presentation, what happening? Cytotoxic CD8 cells are restricted, and after the whole story, when we finish that, we have also learned that another phenomenon that is called MSC restriction, and that because of the MSC restriction, cytotoxic CD8 T cell are restricted to class 1 MSC antigen present on the nucleated body cells, while helper CDT cells are restricted to class 2 MSC molecule. Where they are presented? They are present on the APCs. So what we are going to learn that how these CD8 T cells and CD4 T cells activated. So therefore, we are going to talk in our next lecture how CD cytotoxic T cells and CD helper T cells are activated.